Okay, thank you all for joining us and welcome to the Sepsis Canada Summit Series webinar. My name is Christine Russell. I identify as she, her, and I am part of the communications team at Sepsis Canada. Throughout this series, our research teams will be providing an overview as well as progress to date on their current network projects. We will take about 5 minutes for quick introductions, housekeeping items, and following these, we will begin our presentation. Please feel free to ask questions throughout the webinar today via the chat box. The chat box will be moderated for questions at the end of the presentation, and we will answer them during the final few minutes of the webinar. During the presentation, all microphones will be muted. If you wish to ask a question at the end of the presentation, please feel free to raise your hand and we will enable your microphone to be unmuted. Lastly, just as a reminder, this meeting will be recorded. We will be publishing it on our website in the news and events section for those who will not able to attend today. I would like to acknowledge the Indigenous people of the lands that we are on today. While we meet today on a virtual platform, I would like us to take a moment to acknowledge the importance of the lands which we call home. We do this to reaffirm our commitment and responsibility in improving relationships between nations and improving our own understanding of local Indigenous peoples and their cultures. From coast to coast to coast, we acknowledge the ancestral and unceded territories of all the Inuit, Métis, and First Nations people that call this land home. Sepsis Canada is a nationally coordinated research network with over 200 members and representation from universities and academic institutes across Canada. Funded by the Canadian Institutes of Health Research, Sepsis Canada brings together experts from various disciplines working collaboratively on interrelated projects under an integrated and unified program. Our mission is reducing the burden of sepsis for all Canadians. Sepsis Canada has created 3 teams within our network. And throughout the Sepsis Canada Summit Series, you will have a chance to hear from all 3 teams, which will include updates on their current projects, progress to date, and their next steps. And with that, I would like to now introduce you to today's guests from Team 3, Drs. Michelle Coe and Francois Lamontagne. Dr. Michelle Coe is an assistant professor in the School of Rehabilitation Science at McMaster University in Hamilton, Canada. As a clinician scientist, she also cares for patients as a physiotherapist from the physiotherapy department in the St. Joseph's Healthcare ICU in Hamilton. Dr. Coe is also a member of the Sepsis Canada Steering Committee, as well as a team lead and mentor for Sepsis Canada. Dr. Francois Lamontagne is an intensivist and professor at the University of Sherbrooke, who also holds a master's degree in pharmacology and another in health research methodology. His research activities include clinical trials of resuscitation interventions and knowledge translation activities, such as trustworthy guidelines. Dr. Lamontagne is the co-scientific director of Sepsis Canada, as well as a team lead and mentor for Sepsis Canada. I will now turn it all over to Dr. Lamontagne to share his screen and begin their presentation.
So maybe if you could confirm that you see my screen, Michelle, you could actually begin. Michelle is the one kicking off this presentation. Thanks everyone for, well, thanks for hosting this, Christine and Allison, and thanks for having us, but turning it over to Michelle to begin the presentation.
Thank you so much, Francois. On behalf of the Canadian Clinical Research Network, I am so delighted to share this presentation with my dear colleague, Francois Lamontagne. I'm Michelle Coe, and I'm a physiotherapist and clinician scientist from McMaster University at the School of Rehabilitation Science. In today's talk, we'll give you an overview of the mission and vision of the Canadian Clinical Research Network and also share some of our work to date. We look forward to your comments and feedback and a very rich discussion. And before I start, I'd like to thank our sponsors, particularly Sepsis Canada, for supporting our research and giving us the opportunity to share our work to date. So at the Canadian Clinical Research Network, we believe that conducting high quality clinical research on a large scale is essential to improve the health of Canadians. Our mission is to strengthen the capacity for this research and to improve the coordination of clinical research across Canada. And our overarching objective is to efficiently produce research evidence that will inform healthcare decisions in Canada. We have a multidisciplinary team, and we're very delighted that we have patient partners, scientists, and expert research coordinators. We represent both French and English languages, adult and pediatric research, and clinical and translational research. So why do we need the CCRN? In Canada, we have a fragmented research ecosystem, and we believe that we can be part of the solution to help improve this ecosystem. We're challenged by insufficient infrastructure, and this could be represented through a minimally connected clinical trials ecosystem, whereby each time a clinical trial starts, we need to assemble a team to be able to do the clinical trial. And unfortunately, there is no formal mechanism right now to share that knowledge and infrastructure of human wisdom, but also resources to be able to do clinical research. In Canada, we need the opportunity to be able to build capacity to do clinical research for not only principal investigators, but also the real experts who are helping us answer our questions, the research coordinators. We also have lack of capacity to work between hospital databases and population-based databases. Our current ways of doing research right now also lead to duplication. We have clinical trials that have overlapping trial hypotheses, similar interventions, in similar patient populations. We have challenges in procuring research ethics and regulatory processes. And if we have trials that are similarly answering the same questions for the same patient populations, we then have duplication in the opportunities for people to participate in research, represented by duplication in consent, data collection, and follow-up. And finally, we have limited stakeholder engagement. We see that there are opportunities to improve knowledge translation, stakeholder engagement, and have a unified approach to Canadian research priority setting. However, we can't do that unless we have a sense of what our current status is. So in order to try to improve the research that we do in Canada, we first need to understand what the current status is of research in Canada. And earlier, I described that our mission is to strengthen the capacity for and improve the coordination of research in Canada. So the two main projects we've been working on that we're very excited to share with you are work on monitoring clinical studies across Canada and also tracking clinical study progress across Canada. And we're very grateful for the clinical researchers who have helped us start to work towards this important goal. So if we start to think about what is the opportunity in Canada, we first need to understand where could research occur in Canada. And this is the great work of our research coordinators who have started to identify all of the hospital sites across Canada. And here in this map, you can see that there are several dots, where every dot represents a hospital site. Red dots represent hospital sites where clinical research is occurring, and blue dots represent opportunities for conducting clinical research because they're not yet engaged in clinical research. We might be wondering then, of the red dots, where does research occur in Canada? And if we were to focus a little bit more now on the red dots, the places where research is occurring, we've now superimposed the number of participants enrolled in clinical research in hospitals across Canada. And you can see that there's a large concentration in metropolitan areas and places that are typically affiliated with medical centers. However, you'll also notice that there are some very exciting opportunities where research is occurring in community centers. So how do we get this data? I wanted to give you a little bit of a background into how we ultimately create this measurement of research across Canada. And like everything, it starts with a single study. In this graph, you can see that on the x-axis is month, and on the y-axis, you can see that on the left-hand side, there are the number of participants, and on the right-hand side, the number of sites. The red bars represent the number of hospital sites that this clinical trial is up and running. And the blue line represents the number of patients enrolled over time. So you can have a bit of a window into a multi-center clinical trial to see how we can ramp up recruitment over time and the incremental recruitment that improves by adding sites across the country. However, we might be wanting to try to understand where is this research occurring. And you can start to see that there's a little bit of involvement in Alberta, but most of the involvement occurs within Ontario and Quebec. And then you can start to see where is most of the recruitment occurring. And as I saw that there are both clinician scientists and translational scientists attending this seminar, I'm thinking that you're probably starting to wonder, gee, where are these sites and what types of studies are they doing? And do any of these studies also have clinical samples? So when you start to think about how we can capture data and understand where research is occurring for one study and how well different sites are recruiting for a given clinical trial, you can start to imagine how we can upscale this for multiple clinical trials. And at the very beginning of the pandemic, there was an urgent need to find treatments for COVID-19. And CIHR invested over $250 million in research for COVID. At the time, there were many groups across Canada that applied for these funds. And with the value of hindsight, we can see that there's overlap in some of the studies that were funded. Here is an example of clinical trials where some of the interventions are similar. These studies are occurring in multiple centers. If you look at the far right column at the recruitment per month per site, you can see that this varies by different interventional group. And what I'd like to do is talk you through an opportunity where we can start to systematically collect information about the clinical trials or clinical research occurring across Canada and identify opportunities for collaboration. So here's an example of one study of vitamin C as a treatment for COVID-19. This study, Love It COVID, occurred in 13 sites, enrolling about 2.4 patients per month per site. Another study, REMAP-CAP, also studied vitamin C. This was occurring in 35 sites with a recruitment rate of about 1.3 patients per month per site. If we look at where both of the studies were occurring, you could see that there is an opportunity to potentially leverage research infrastructure, identify duplication, and with hindsight, could we coordinate the research better to answer the question more quickly? So these are starts of the types of things that we need to be able to measure in order to improve research across the country. So in summary, I've given you a brief overview into the type of clinical studies and clinical study tracking we have conducted over the last few months. Ideally, we want to be able to improve the opportunity for Canadians to be able to access clinical research. If we think about the opportunities, you're probably thinking about, are there opportunities to marry biobank samples or translational research with these clinical studies? And I'm delighted to hand things over to Francois, who will speak to these next points.
Thanks. Thanks very much, Michelle. I'm going to try to, I don't know if I speak half as eloquently as you just have, I'll be quite happy with myself. So as Michelle explained, this work that we undertook under the auspices in part of Sepsis Canada. Sepsis Canada was one of the inaugural sponsors for this work. Within the Team 3 activities, we, as Michelle explained, this work is designed to build better infrastructure for clinical research, and it certainly is an important motivation. And up on the screen, you see the core values that underpin this work. We believe that science should be the foundation upon which we make decisions in health. We believe that there is this is too important for this for these challenges to be addressed solely by scientists. We believe other stakeholder groups like the entire entire population of Canada, health system administrators, funders, and other groups, really must be at the table to help structure and restructure and improve this way of conducting clinical research. We strongly believe that this is so important that every group in Canada has to be adequately represented. And if the infrastructure isn't allowing every group to be well represented, then it needs to be fixed. And lastly, this could be done quite a bit more efficiently. And so Michelle has spoken very eloquently about this. And most of the time when we describe our activities, we talk about it in a very classical sense, in terms of clinical research. People think of clinical trials and cohort studies mostly, but since this was conducted on behalf of Sepsis Canada, and seeing how translational research is real crucial for Sepsis Canada, I wouldn't want you to leave today thinking that this leaves out the entire domain of translational science. It is indeed a very important motivation, and these core values are as relevant to the translational research endeavor. And so in this part of the presentation, I'm here to explain briefly how we believe this kills two birds with the same stone, and also has the potential to really be a bit of a game changer for basic science and translational research in Canada as well. So for the purpose of the discussion today, understanding that translational research means a lot of different things, I would like to focus on what is in this red rectangle, and we'll be talking about translational research in the sense of this crosstalk that could and should happen between basic science and human and clinical research. Importantly, this crosstalk has to be bidirectional. Everyone understands that basic science can and should inform the design of clinical studies, but the reverse is also true. Clinical research can and should advance knowledge, and particularly our basic science understanding of human illnesses. At the bottom of the slide, there's just one of many examples where if you do this correctly, the clinical research infrastructure can contribute greatly to advancing knowledge, and recently colleagues from Edinburgh benefiting from a biobank infrastructure in the UK have shed light, or have published a study in Nature shedding light on the some genetic factors that are important in understanding critical illness in COVID. Now, these colleagues were able to do this because a long time ago, quite a few years ago, there was a decision made to invest in a very ambitious project, a UK biobank, whereby data and biological specimens were to be collected prospectively over time in order to answer future research questions. So this is in a way the exemplar standalone biobank, what we'll define as a standalone biobank, and it's proven instrumental in answering many important questions, and it will continue to do that in the future. But this is an ambitious project, and it's onerous. And it's onerous, perhaps not in the ways that we think it's onerous. I mean, obviously, a number of freezers and a lot of material infrastructure has to be bought and installed across the country. But what is really onerous about work like this is that people, research staff, have to prospectively identify potential participants, seek consent, obtain consent, and collect both data and specimens. We can always figure out ways to collect clinical data from electronic records, although it's not always as easily done as said, but you need someone to draw blood. And so, whereas this model of a standalone biobank really has a lot to contribute, we'd like to discuss this operational definition of a biobank, which is simply the combination of data and specimens that are matched. And we'd like to discuss an alternate model, making it possible to build a biobank, but not as a standalone project, rather as a project that is nested within other clinical research endeavors. So I'm using the same maps that Michelle showed you a few moments ago. If you think of it, every clinical study, I mean, Michelle's right, we are doing everything we can to increase the number of red dots. So we'd like those blue dots to turn red. But already today, there are intense research activities occurring across many sites in Canada, and every clinical study being conducted in Canada collects clinical data. So you've got at least one part of that equation already covered by those clinical studies. And I would go as far to say that the clinical data collected in the context of a clinical study is actually very high quality. It's rather, there's a lot of it, and it's, a lot of very smart people spend a lot of time making sure that that clinical data is of the highest quality. But on top of that, a lot of those clinical studies also draw blood and collect samples simply because those clinical studies are designed to answer clinical questions that require laboratory analyses. And some of those studies go even further and preemptively seek consent from participants to store said data and specimens beyond the study itself. So beyond those pre-specified research questions, with a view to answer future research questions that weren't posed when the study was designed. So basically, some of those studies have nested biobank effort within the study itself. Now, the CCRN is a relatively new program, and thanks to the support of all the colleagues and investigators who have agreed to share information, we've already built a pretty meaningful portfolio of studies. A lot of those studies focused on COVID, but we haven't limited the scope of our activities to COVID. A lot of those studies are about sepsis or renal replacement therapy and organ donation. And this is a list of clinical trials only. So there are a number of observational studies in our portfolio and standalone biobanks, but these are just clinical trials. And if you were to add up the number of participants enrolled across those clinical trials, so again, only the clinical trials, and if you started counting from January 2020 to today, you would find that almost 9,000 participants have been enrolled across those trials, and that's just a really small minority of all clinical trials conducted across Canada during that period. So assume that, assuming only 30 or 40% of those individuals who consented to participating in those trials also consented to biobank activities. So if they also consented to allow the scientists to store the data and the biological specimens for longer than needed to answer the clinical trials research questions, well, already immediately you are looking into potentially a very sizable, albeit decentralized, national biobank infrastructure. So what we have to sell here, or what we like to convince you, or what we'd like to share basically, is this vision of a real opportunity for growth, whereby every clinical study that is already collecting clinical data and quite possibly collecting biological specimens could share information about what is being collected, and maybe preemptively decide to obtain or seek consent for future research use of data and specimens. And then once that information is available, then it becomes possible to collaborate. And when you think of it, this model wouldn't be perfect, but it has the advantage of being extremely efficient, because the data and the specimens that we're talking of right now, understanding that what is onerous, what is very costly, is collecting those samples. All of that's already been paid for by the funds allocated and entrusted to clinician scientists to conduct the clinical studies. And so it's a highly efficient model, because basically you don't have to reinvent the wheel and start from scratch collecting data and samples. It's also a model that is very nimble, because most standalone biobanks, even though they have a vision of being relevant for future questions that aren't posed when the infrastructure is being built, they're still built in a specific context. During the pandemic, biobanks might have been created, but with the view mostly of answering research question about COVID. And if the research priority in Canada 6 months from now becomes organ donation, well, those are big ships to turn around. They may not be well suited to address research questions in other areas. Meanwhile, all those clinical studies about organ donation and in other areas, they're happening. And if the data and specimens that are being collected are available for biobanking effort, a collective biobanking effort, these multiple biobank hubs are a lot more nimble, and they're available rapidly to answer questions in areas that aren't already a priority, but that could become a priority in the future. Now, the first step is making this possible. And to show you how we're doing this within the CCRN, I'm going to show you the back end of our database. So whenever our team collects information about studies, not only are we collecting information about the research question that is being addressed and the recruitment data, the clinical data that's being collected, we're also asking about the type of biological samples that are being stored. So the first question is, are you collecting any samples? And if yes, are you getting consent for future research use to answer unspecified research questions? And if the answer is yes, we go a step further and ask about whether consent was obtained for genetic analyses, because the legal and ethical implications of genetic analyses obviously are different. And then we ask about whether biological samples were collected at a single or multiple time points, and which time points we're speaking of, and so forth, until we learn pretty much everything there is to learn about the biobank that is being created either as a standalone project or nested within a clinical research project. And so of the relatively small number of studies included in our portfolio thus far, we're already in a position to tell you that there are 16 biobanks in this portfolio. Some are standalone projects, and some were nested within clinical trials and cohort studies. And at the right-hand side of the slide, you can see a very, very brief summary of the types of samples that were collected. And also notice that a lot of, many times the different studies collect similar samples. So many biobanking activities include, plasma or blood collected in an EDTA tube. And so this overlap, this redundancy actually is a good thing, because it means that there is a real opportunity here to join forces and collaborate where there is an appetite to collaborate. Already with the information that we've collected, we're able to share reports. So if some of you, Sepsis Canada members and scientists are interested in a study, you could query the website that we're designing and which will be available very, very shortly, and ask about, how many biobanks are out there, what was the population of interest, was consent obtained for genetic analyses, at which time point, alongside which other types of samples. And remember, we have very granular information about the matched clinical data in each of those studies as well. So our vision is that by making it possible to collaborate, there will be collaborations. There will be these spontaneous collaborations, and there already have been. We're still in the very early phase of this CCRN adventure, but already I'm allowed to share this example because I was personally involved, and I I received authorization from Dr. Jim Russell from Vancouver to share some of this information with you. I think Jim is even with us today. So this is like a brief story or brief success story of a collaboration between Jim's ARBS Corona program and the OVATION-65 trial led by Neil Adhikari and I. Dr. Russell was funded by CIHR to undertake the ARBS Corona program, which includes a trial, but also includes an observational study intended, meant or designed to better understand the impact of the or the role of the renin-angiotensin system in shock due to COVID versus other types of shock. However, Jim was funded, during the pandemic to enroll patients with COVID and didn't necessarily have the resources to recruit patients with non-COVID sepsis. At the same time, across Canada, Neil and I had enrolled a significant number of patients in the OVATION-65 trial. We enrolled patients with non-COVID sepsis who were randomized to receive, vasopressors one way or another, but basically all patients in this trial did constitute, candidate sepsis controls for Jim's ARBS Corona program of research. And so by virtue of collecting information about the type of data that was collected in both studies and the type of biological samples that was being collected, it was possible for Jim's colleagues and collaborators to see that there was, something that we had that could support their research program, and they requested access to the data and to biological samples, and we were able to share some of those samples and conduct, the analyses that some of which appear on the slide here. And that are currently being prepared for submission to a peer review journal in the near future. So one idea is that by virtue of just making information about the Canadian clinical research portfolio spontaneously, some collaborations will arise. It's difficult to know, which of these many hubs, if each of these smaller dots represent a clinical study with data and samples. It's hard to tell who will collaborate, but any collaboration is, brings added value. But we can then start thinking about ways to make this more systematic, about ways to incentivize these collaborations. And you could imagine, for example, that if someone is seeking resources, public funds to conduct, to collect data and biological specimens, a first step could be, requesting that they look across the Canadian clinical research portfolio to see if, useful data and samples are not already being collected, somewhere, which would allow us to perhaps save a lot of those, limited resources that we have for clinical research, to avoid duplicating the work of getting consent, collecting data, collecting blood without always reinventing the wheel. And in parallel, perhaps it might be expected of trialists such as myself, who are, who have the privilege of receiving funds and the privilege of using public infrastructure to conduct, research that they lead. Perhaps trialists and clinician scientists might be expected to store some biological samples or systematically seek consent for a nested biobank, and then store some of those biological samples for others, like for future research questions posed by others. And so there are various ways in which you might envision a way to incentivize and even sort of make this type of collaboration, well, not only possible, but to the, at some point mandatory. So again, the whole point here is that research is important. It's important for quite a few groups beyond just the scientists. And so we are going to strive to make other groups, the public, patients and families and health system administrators, understand that they all have a stake in how clinical research is conducted. And of those groups, basic scientists have a lot to gain, gain from clinical research infrastructure that is more representative, more sustainable, and more efficient as well. And so that pretty much concludes our pitch. We wanted to leave some time for discussion, and I'd like to just thank you and let you know that you can contact us if you'd like to continue this discussion beyond today's webinar. Thank you.
We'll now open the floor for questions. I don't see any in the chat box, so we can.
Yeah, I just put a question in there.
Oh,
Jim.
sorry.
It's more of a comment than question. Can you hear me okay?
Yeah, go ahead, Jim.
Yeah, thank you very much for organizing this. Very, very interesting for me in the periphery, way out here in Vancouver, to see the success of all the work you're doing, Michelle and Francois and team. And I think Francois said it well. We had a meeting of minds and energy to collaborate, and it's coming along well. It's actually going to be a very, very nice manuscript that will contribute to the understanding of sepsis and COVID-19. I wanted to make a general comment that I think is very, very important at this time. You highlighted the hundreds, literally hundreds of millions of dollars of research put into COVID. And as we all know, COVID is sepsis due to one particular virus. So just as many of us pivoted from sepsis to COVID because we have so much sepsis expertise, now we have an opportunity to pivot from COVID back to sepsis. And what I mean by that is another specific example. The study that Francois and our team and his team did together shows us that there are some similarities and differences in the renin-angiotensin system dysregulation in COVID and sepsis. So how was that going to affect, well, a treatment? It could affect the use of these drugs during COVID. They're being tested in trials. But it also can address specific clinical questions we often have in the ICU on the ward. Should we continue those drugs in a critically ill patient who's unstable? Should we continue them on the ward patient in a patient who's not unstable but might get unstable? And some of the biobank efforts can help address that by looking at risk groups. And so what we're looking at now, and I'm going to invite another collaboration with Francois because we got our sixth grant just about 2 weeks ago for this ARBS Corona program. We're going to expand, and now we're organizing to do proteomics, an immune marker panel, and a cytokine panel in COVID-19 patients acutely at 3 months and 6 months. And I think it would be very helpful to have a control group in sepsis patients, because I think we'll need to have these signatures from the biomarkers that help us determine, in this example, which drugs we should continue or not. And ultimately, would we even want to do trials in sepsis? So, Francois, I don't know what you think of that comment and that invitation to collaborate yet again. I'm very, very pleased with the way it's worked out. And as I said, with this extra funding, we're now in a position we're moving along to do these other biomarker measurements in the COVID patients. And by the way, we have about 5,500 blood specimens now in about 2,500 COVID-19 patients across Canada and France.
Yeah, so there are a number of hands that popped up, so I'll let other people speak, but just because you asked me to respond, I think, so congratulations on obviously the grant and all the work that you've been doing. And about the model, I think it's and so it's great that you were able to attend this today, because the example I provided is it's like we're super happy that we did this. We did it on a small scale, but you can imagine that you have the opportunity to do this on a much broader scale with a large number of sepsis studies, clinical studies in sepsis. And I'm sure that not all of them would have the type of data and specimens that you need, but perhaps a significant number do. And there is and so this effort that Michelle and I are leading is to make it possible for this to snowball, basically. And I think it's so great that you're doing this. Great model, great to reproduce this, and this is, I think, the network to do it with. I think Sepsis Canada, this is one of the many things that Sepsis Canada really wants to do, is allow these type of ambitious collaborations that might not have occurred otherwise to occur now. And I'd like also to emphasize your point about, there was, so this collaboration between our groups meant that you made good use of data and specimens that were collected before anyone knew anything about COVID, right? The OVATION was designed before, so that became useful later on, even though we hadn't anticipated what our future research needs would be. And now you're emphasizing that what we learned during COVID, because there were good reasons to focus on the renin-angiotensin system, but that could very well play. It could be very meaningful and important for all sorts of different kinds of sepsis, right? If only because a lot of the patients with sepsis are already treated with ACE inhibitors and ARBS, and so it becomes extremely relevant. And again, the grant that you got for COVID wasn't, you designed a project that wasn't intended as something that would solve the roles of RAS in every other instance of sepsis, but because you collected those samples, because you might be willing to share information about the samples and entertain collaborations with other Sepsis Canada members, then it becomes possible to continue to build, to just make good use of that investment that Canada made by when they funded your group. It continues to pays off, basically. So I think that it's so challenging, to build the one standalone biobank for the entire country that will be set up to answer every future foreseeable question, that at the very least, if we make it possible for stuff that we're already paying for, we're already buying all of this clinical research, there's just a little bit of, of this little bit of extra effort of, seeking consent for storage and data and specimens. It's just a very, very efficient way of building on the research that we're already doing. So yeah, I'm going to stop, and I think Brent was first, and then Allison raised her hand.
Yeah, thanks. I highly encourage this activity. I have been trying for over 20 years to get clinical trialists to collect specimens in an appropriate way that can be used in the future with consent for them to be used for future questions. So I think it would be really helpful to help the trialists on models of consent to do that, methodologies to collect the samples to make it economically feasible, and a cooperative method. Jim and I have also collaborated on some sepsis stuff, and I actually think it makes it very, very useful. There are, for example, we have a tissue bank in Scepter for sepsis. I know that Hamilton has a tissue bank for sepsis. And so there's lots out there that people can be using.
Yeah, so responding to that, again, thanks for your comment. And just before Allison comments or asks a question, so you mentioned that you've been open for business with trialists for a while, right? And have had, I don't know, moderate or a lot of success, but I guess you'd take more, right? And you'd be willing to do more with and I think that is sort of a challenge that we've identified, this fragmented effort, right? The lack of engagement across different stakeholder groups. The values that underpin the CCRN, underneath it all, we think that research is critically important, and being able to lead clinical research is really a privilege, right? Because you are using public infrastructure. All of this research is happening in the hospital system, and it gets somewhere there is a public infrastructure that is making it possible for someone like me to conduct my trial. And if the rest of our discussion, and I think Brenda is on the call and other people, from the trials group and the clinical trials network, and if this other part of the discussion whereby we're going to try to improve this infrastructure to allow us to build bigger, better trials, conduct them more efficiently, well, if this investment is, happens in clinical research, it should be a shared infrastructure. So a trial is benefiting for the current infrastructure, and even more so this improved infrastructure. Their access to this infrastructure could be conditional upon doing something for the other stakeholder group. So I think that a trialist doesn't own the research that they lead. They're doing this on behalf of a group of Canada, right? Citizens, patients, and so you might expect that citizens, patients, and the public health system should have those groups already have a stake in the research agenda, the portfolio. But basic scientists are an important stakeholder group, and they have a stake in this too. So it might be expected, if trialists like me, have the privilege of using this, these funds and this infrastructure, that they are going to set something aside for the greater good and to support the advancement of basic science as well. So I think all of a sudden, if we invest in this clinical research, then there's leverage to get from it what we expect to get from it, and that includes basic scientists as well.
Thanks.
Allison, yeah, go ahead.
Yeah, thanks, Michelle and Francois, for a really great overview of all of the great work you've been doing in the midst of the pandemic and since we got the network set up. Francois, the one other idea that's come forth in the network is how we use this clinical data and who's enrolled in those studies to actually better understand the population of health of the diseases that we and the people that we're enrolling in trials. So the other consenting issue for the trialists around the table is that we need to consent to be able to store and keep that data. Team 1 is very interested in using clinical trial metadata to add to their metadata catalogs that are part of understanding the population health of sepsis and other diseases that affect people who come through our ICUs or are involved in clinical trials. And so it does require, and I know Michelle and Francois have both heard about this, but it does require some pivoting on the consent forms, that we have the ability to share and store the data from those trials, at least to know what trials we're enrolling, what sorts of patient into a metadata catalog, and how many of those patients were, for example, septic. And it could apply also to those observational studies in which we're collecting biobank samples on septic patients or otherwise. So I think there's good synergy between the work that's happening with the CRN and the work that's happening in terms of the metadata catalogs and the storage of data for Team 1. But it does require all of the trialists, and Brenda knows this because we've been talking about it at the trials group as well, how we change our consenting process so that we can store that data for other people to study who's been enrolled in trials. So again, thank you very much.
Thank you. Yeah, when we mentioned preemptive efforts to store data and samples for future use, this storage, this encompassed everything, storing, but sharing as well. Michelle and I aren't we're not actively trying to change the way people do things. At this stage, we're just describing what's already happening, understanding that already there's at least a subset of those clinical studies that have already that they're already getting consent for storage and sharing and future use. And by making this information public knowledge, then it's possible to collaborate, and it's possible to use what's already being done. And then building on that, the next phase would be making this effort of storing and then getting consent or offering participation to contribute to biobanking, including, you know, sharing data and specimen, making this more systematic. We might want to increase the number or the proportion of studies that do this, but as a first step, just making the information available about what's already happening is already, in our view, a great significant contribution.
I agree, and I'd like to add that to build on Brent's comment, how do we bring people together to understand how we can complement the other's research? So in addition to having the consent forms, the clinical trialists might not know what is required for biospecimens or build budget into that for clinical trials. So pulling together experts from the different groups to be able to prospectively plan a study around or prospectively plan our studies and organize and coordinate all of that, because I can just also think in addition to getting consent for future use of samples, consent for being able to link to population health databases. So there's this bigger infrastructure that we can be thinking of to improve the efficiency of research across Canada.
Jim has another comment.
Yeah, thank you. Those are excellent comments. I think the other opportunistic timeliness right now is that some of the presentations and grants and other things I'm writing now, you know, there probably isn't an adult in the world who hasn't now seen the inside of an ICU and what a patient on a ventilator looks like, literally. And before, we all know that before this happened, when you talk to even friends and family and you explained you're an intensivist, they say, which emergency room do you work in? You know, there's just this incredible misunderstanding of what intensive care is and what we do. And there's no knowledge of sepsis. That's getting better, but it's far from common knowledge. So I think there's a real opportunity to take the benefits that we've learned through the COVID pandemic scientifically, both at the population, biobank, trial level, and really almost bombard the granting agencies now with grants to look at sepsis. You know, our time is now. In a year, 2, 3 years from now, unless the pandemic continues and continues, sepsis will be forgotten. Long COVID and long sepsis will be forgotten. So I really think there's a very strong opportunity. So the specific example, we're now pivoting to community-acquired pneumonia on the same model as our first cohort, using exactly the same protocol so that we can have a control group of non-COVID community-acquired pneumonia. And that was the one that was granted from CHR a while ago. I think for all of us, there's this opportunity that you're pulling together groups for collaboration. And that's what the reviewers love to see for some of these bigger projects. And I think the topic of the day, I think for the next couple of years, needs to be sepsis, septic shock, community-acquired pneumonia, because I'm hoping reviewers will see that it's very, very relevant, as Canadians do. Anyway, thanks for all these great comments and discussion. Very helpful.
Anybody have any other questions? Comments? If there aren't any other questions, on behalf of Sepsis Canada, we would like to thank you for joining us today. And of course, Drs. Coe and Lamontagne for their presentation today. Our next webinar is April 22nd at the same time, 12 p.m. Eastern to 1 p.m. Eastern, where we will be joined by Dr. Gina Parsons-Lee from Team 2, who will speak about their progress on their project of creating a culture of sepsis awareness through advocacy, health literacy, and knowledge transfer. I put the link to register in the chat box for anybody that wants to register now while it's in the chat box. I will also send it out via the newsletter. You can follow us on Twitter, Facebook, Instagram, and find it there. You can also find the contacts on our webpage to contact Drs. Coe and Lamontagne if you want to reach out to them further about the presentation today. Thank you all for joining us.
Christine,
Thanks, take care.
Oh, yes, Alan.
quick question. I obviously got the time wrong.
Oh,
But
I know, I just let you in.
I saw it was recorded. Can I see the recording someplace?
Yes, you can. I'm going to put it on the website. Probably in the next couple days, you'll see it on the website, but I'll send you the link, okay?
Okay, thank you very
Yeah,
much.
you're
Sorry
welcome.
about that.
Yeah, that's okay.